Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture I am going to talk about regulatory T cell or T regulatory cell. We also call it suppressor T cell. Okay, in the previous video lecture, if you guys remember, we talked about helper T cell in detail. We talked the types of helper T cell, we talked the function of each helper T cell and cytokines which are released from the helper T cell, each and everything in detail. And we also talked that helper T cell lecture is actually related to T cell biology. And if you guys remember, in T cell biology, we talked about the T cell biology overview. We talked about T cell receptor. We talked about the types of T cell. Means these are 3 or 4 videos we talked about before, before this video. So we talked almost 4 video lecture which are related to T cell biology. So these video lecture are very important for you guys to know, to understand about the basic of T cell biology, and moreover to know about the T cell types. Now in this video we are going to talk about T regulatory cell in detail. So let me remove this portion and let me start. Okay friends, so now let me write cell. Okay, so T regulated cell or suppressor T cell, it is again the type of helper T cell. Okay, but we didn't talk to this T regulated cell in helper T cell video lecture. Now T regulated cell is a type of in this video lecture we are going to talk about the introduction of T regulated cell, development and activation of the T regulator cell, structure of T regulator cell, and moreover we are going to talk about the function of T regulatory cell. First of all, let's talk about the introduction of T regulatory cell. Now T regulatory cell is a type of helper T cell which are activated at that time when there is hyper amino cell regulation or when there is hyper amino cell activation occur. Then T regulatory cell will activate to control that disorder. Actually it is a kind of disorder. Okay, I will explain it later. So this is a little bit a brief introduction that is related to T regulatory cell. Now let's talk about their development and activation. Mainly T regulatory cell are developed from the bone marrow. If let's suppose this is the bone marrow, as you can see. Okay, so if let's suppose this is the bone marrow, inside the bone marrow the T regulatory cell are developed. Now how? Inside the bone marrow we have a cell which we call that stem cell. We call that hematopoietic stem cell. From this hematopoietic stem cell, different cells are amino cells are formed. But this hematopoietic cell are converted into lymphoid progeneratory cell. This is the lymphoid progeneratory cell, and from that lymphoid progeneratory cell we get T cell. Okay, this T cell is actually regulatory T cell, but how this T cell is converted into T regulatory T cell? Now once the T cell are get developed, so then this developed T cell are migrated to the thymus area which is present at that area, as you can see. So this T cell once reach to the thymus portion of the body. Let's suppose this is the thymus portion. Although this is not the exact structure of the thymus, but somehow it will be look like in this form. Now once it reach to the thymus area, then inside the thymus this T cell are get mature. Means there are some macrophages or antigen presenting cell that guide the T cell that what you need to do in the immune system. Okay, normally what we have the macrophages and dendritic cell, as you can see. Normally we have dendritic cell. So let's suppose this is the dendritic cell. And for the maturation of T cell, they can present a kind of antigen to their receptor. Inshallah, in upcoming video lecture I will explain the development of T cell in detail, but this is just the overview or basics about the T cell development, due to which we will get the idea about the T regulatory cell. Okay, now this antigen presenter cell presenting a antigen fragment to just guide the T cell, that what you need to do. The T cell will just bind here. This is, let's suppose, the T cell receptor. This T cell receptor will just bind here, and after that this T cell will decide to produce other T cell, or we can say helper T cell, cytotoxic T cell, effector T cell, memory T cell, T regulatory cell. Among them T regulatory cell is also produced. But what actually going on, this dendritic cell or this antigen presenting cell will, once they are attracted, then this dendritic cell will guide the T cell that you need to produce those T cell which are against this antigen portion. If let's suppose this antigen portion is virus, so then this dendritic cell or antigen presenting cell guide the T cell that you will convert yourself, or you will attract the cytotoxic T lymphocyte. After that this T cell will attract the cytotoxic T lymphocyte. Okay, so from there the T cell will get matured, and if this T cell doesn't perform their function, so then it will be rejected. And when it perform their function, let's suppose it perform their function and it producing the other cells like memory T cell, and they are also producing like other cell which we call that effector T cell. Effector T cell. This effector T cell will further produce cytokines. Each cytokines will then produce, or each cytokine will then attract the what we can say macrophages, or each cytokines. This effector cell will actually activate the other cells. If the process is going on, then these cells will be accepted and positive selection will occur, due to which this cell will be migrated to the bloodstream and perform their function. Now it's matured. But if it doesn't produce memory T cell, it doesn't produce effector T cell, the effector T cell will doesn't produce the other rest of the cells T cell, then what happen? This cell will be rejected, and inside the thymus these T cells are die. Okay, so this is how the T cells will get developed. Now once it means matured, then after that this effector T cell will produce we can say that cytotoxic T lymphocyte, natural killer cell, and T regulatory cell is also helper T cell. Okay, so we can say that from that portion the T regulatory cell are actually mature or developed from that portion. Okay, so this is about the maturation and development of the T regulatory cell, that how it will get developed and mature. Inshallah, in upcoming video lecture I will explain the activation. Moreover, I will also explain the development and the maturation of the T cell in detail, okay, in upcoming video lecture. So this is a little bit about that how the T regulatory cell will get developed and activated, or we can say that mature. Now let's talk about the structure of the T regulatory cell. Normally this is the structure which I am drawing here. It is not the exact structure. Main thing which are present in the T regulatory cell are is. So let me draw a rough diagram of the T regulatory cell. Let's suppose this is the T regulatory cell. A rough diagram. Okay. On the surface of each T regulatory cell we have different kinds of receptors, like T cell receptor, which is very common. So let's suppose this is a T cell receptor. Let me write TCR, T cell receptor. Moreover, they have another co-receptor molecule which we call that CD4 positive receptor, and moreover they have another receptor which we call that CD25. Let me draw CD25, or we can say interleukin 2 alpha chain. Moreover, they have another receptor. Let me draw here with this black marker. Okay. Now this receptor is called CTLA4. Okay. And inside this T regulatory cell, what we have the nucleus portion. As you can see, inside this nucleus they have the genetic material, which we call that FOXP. This is actually not the real, but this is somehow the T regulatory structure. They have all these main component and their genetic material. Now, this is the structure. Let's a little bit talk about this genetic material because it is very important to understand about the T regulatory cell. You know, this genetic material, if we talked about, so in this genetic material, they have two conserved sequences, which we call that conserved non-coding sequencing. Let's suppose like that portion, which we call it CNS1 or CNS2. We call that, you know, conserved non-coding sequence 1 or conserved non-coding sequence 2. These two sequences actually enhance. Okay, actually these are the high level of transcriptional factor. Due to this way, they will, you know, produce and transcribe. Due to this way, this T cell, let's suppose this is actually the T cell, then after that it will be converted into T regulatory cell, which is done by this genetic material. And after that, they will perform their function. If these things are not present, then if these genes are not performing their function, if these genes are not present, then this will be the T cell and it will be not the T regulatory cell. So for the conversion of T regulatory cell, these genes, expression of these genes are very important. Okay. Now let's talk about the function of T regulatory cell. So this is all about the structure, activation, development and introduction. Now let's talk their function. You know what we have normally, we have the macrophages, let's suppose. You know, if there is a disease condition, so let's suppose we have a macrophages. If they engulf the bacterial cell or viral component, then they will digest it, and after that they will present or expressing the portion of that organism at their receptor, which we call that MHC2 molecule. So let's suppose this is the portion. After that, what happen? They will secrete different kinds of cytokines, which we call it pro-inflammatory cytokines. And among this pro-inflammatory cytokine, we have interleukin 6, interleukin 2, interleukin 4, and we have also, you know, tumor necrosis factor and some other cytokine as well. But these are the common cytokines which are produced. Once these cytokines are produced, then, you know, a lot of T cell are attracted. For example, interleukin 6 will attract the helper T cell, interleukin 2 will attract the cytotoxic T cell. Let's suppose interleukin 4 will attract the helper T cell 2, and tumor necrosis factor will attract natural killer cell. So we can say that a lot of cytokines are produced and a lot of cells are attracted. Although each cell can, you know, destroy the pathogen or kill the pathogen, means they will first recognize and after that they will kill the pathogen. Moreover, they will also kill these macrophages. You know, sometimes macrophages, a lot of macrophages are also attracted. When all the amino cells, we can say, when all the amino cells are attracted, a lot of amino cells are attracted, which is abnormal, you know, process. Then what happen? Although each amino cell can destroy the pathogen, but after that, each amino cells are, you know, present in a large number. And if there is no function of each amino cells, then what actually they will do? They will cause the destruction of the body tissue. They will destroy the body tissue. Why? Because it is present in a large number, and if there is no job for that T cell, then what they will do? Definitely they will, cause the destruction of the of our own body tissue. So that's why, this is sometime happen, and we call that process autoimmune disease, if you remember. Okay, so we call that autoimmune disease. So in autoimmune disease, all of you better know, a lot of hyperimmunity will occur, or hypersensitivity reaction will occur. A lot of amino cells will get activated, and due to this way, each amino cells will, against our body, means they will cause the destruction of our host cell, and due to this way, some of portion of our body will got damaged, which is done by this autoimmune disease. This is abnormal process. Then what happened at that time, interleukin 2 will secrete it then at that time a lot of T regulator cells are also secreted. But some of the T regulator cell will attracted with this macrophages. There is not only one macrophage. Here I draw just one macrophage, but there is a lot of macrophages, and they are representing the antigen portion. Then a lot of T regulator cell will come. Let's suppose here I will draw one of the T regulator cell, that what happen normally or in disease condition, then they will recognize this antigen. So let's suppose this is the T regulatory cell. Moreover, they have also this CD80 or 86. And with this T regulator cell, they have the co-receptor, which we call that CTLA4. So these are the co-receptor, means CTLA4 and CD80 respectively. When these binding occur, when the recognition are done, then strong recognition are happen at that condition, as you can see. After that, what happened? This T regulator cell will secrete two types of cytokines, which we call that interleukin 10, and moreover they will secrete transforming growth factor beta. So these are the two cytokines which are released by this T regulatory cell at autoimmune disease condition. After that, what happened? This interleukin 10 will come contact with all the macrophages or all the antigen presenting cell which are activated during autoimmune disease. So all interleukin will bind with the antigen presenting cell, and once it binded, so then they will give signal to the nucleus. Let's suppose this is the nucleus. After this signal, nucleus will stop secreting these pro-inflammatory cytokines. So pro-inflammatory cytokines will be not secreted. Due to this way, other cells will be not activated, means other T cell, regulatory T cell, or other antigen presenting cell, or we can say other immuno cells will be not activated, because once it's secreted, so inflammation will also occur, inflammatory reaction will also occur, reaction will also occur. But if it blocked, so then no inflammatory reaction and no autoimmune disease will occur. Or we can say that the autoimmune disease are under the control. Sometimes they have another factor which we call it cytokine, which we call it transfer growth factor or transforming growth factor beta. This kind of cytokine will affect on the T regulatory cell. Once they affect on the T regulatory cell, so then T regulatory cell, there is means these kinds of genes. So these genes are silent. If these genes are silent, so no more T regulatory cell are secreted. So we got a point that once during autoimmune disease, when T regulatory cells are activated, then they will stop the rest of the other cells activation. Okay, so this is all about the function of T regulatory cell, that how actually they will perform their function. It's all about the T regulatory cell, their structure, introduction, activation, development, and function as well. I hope you guys get idea about that. If still you have any kind of question in your mind, I will try my best to give you answer as soon as possible. If you like this video, so please hit the like button and share this video. Definitely subscribe my channel to get more videos lecture which are regarded to T cell biology. Thank you so much for watching.